Zoom mode up here. And so we can't share it to get the full screen. So somebody going to get up and move closer.
That's as big as I can do.
Well, okay. Terrible, so. And. All right, so while you're fiddling with that, so we're very fortunate to have Margaret also on our advisory board. She is the director of the newly created Physics Frontier Center for the Center for Living Systems up in Chicago. She's a professor of physics. I think you also have joint appointments in a couple other places of molecular genetics and cell biology. And so I asked her if she can give us a little bit of an overview about what's going on up there, but I know all the center directors that have come so far, they don't mind telling you about the center, but they want to tell you also about the research.
Sorry, I wasn't sure if the instructions came from you or I suggested it. That's why.
I'm the feeling, I'm just really happy that you guys came. We got an enormous amount of wisdom and insight from you all, and you can do whatever you
Awesome.
want.
Okay. Sorry, you're stuck with me. No, thank you. I had a really great morning here too, and it's really fun to learn about the things going on here. I do, as all of us do, wear many hats these days. I did want to take this opportunity to put a plug for a new journal that I have helped launch over the past 2 years. So this is PRX Life. From the American Physical Society. So it's a nonprofit journal. We've been open for submissions for 18 months. We've been trying to incorporate collaborative peer review so that with interdisciplinary work such as ours, you get reviewed by peers who understand your work, and if there's really differences of opinions, they can hash it out, sort of like an eLife Light model. So please submit your work there. The APC fees are probably going to be waived a little longer until we get an impact factor, which seems like it will be good, right? So that's so I think you're going to have another 6 months at least of doing this. So please submit if you have any questions. You can talk to me about. But all the work being done here definitely fits within what we hope to publish in PRX Life. Okay, so going back to my group, over the past decade or so, I've been really interested in understanding how cells work in collectives. So most of the work in my lab these days focuses on tissue scale phenomena. We've been working on epithelial tissue dynamics, which I think are, really fascinating tissues. They're their most predominant cell type. They form barriers between organs in our body. And they also guide the shape of organs, right? So in epithelia, you have these competitions between control of cell proliferation or cell number and cell cycle and the overall shape of the organ you wish to achieve. More recently, we've gotten really interested in endothelial tissue, which are the cell types that form the inner lining of your vasculature. Here, this is a more leaky barrier. It allows for immune signaling to transpire between the bloodstream and the background tissue. So we've been interested in understanding the morphological processes that allow to modify the barrier function of endothelial, especially in response to different kinds of mechanical cues. Now at heart, we are interested in these questions, how the cell builds physicochemical structures to control the shape and adhesion and motility. I should have probably made this animate. So you think of the cell as a black box, you have a cell have an external force being applied to the cell, and right out of that magic black box of the cytoplasm and cytoskeleton, right, you have things like movement or migration or proliferation, right? We want to look inside the hood, like many of us here, and see all the structures that are in here. So from my perspective, I'm very cytoskeletal biased, right? All the mechanical information processing that goes on from the cell mechanobiology really has to traffic through the cytoskeleton, right? And I'm pretty focused on the actin cytoskeleton, although I acknowledge that's a pretty big bias to have. So we think about, how cells, the nature of cell-cell adhesions between each other or adherence junctions, how cells are attached to their extracellular matrix or focal adhesions, and of course the the connections, the mechanical connections between these adhesion sites, which is predominantly the actin cytoskeleton. And I'm going to even make that a little bit more focused on the actin-myosin cytoskeleton. So these are the polymer networks that build active contractile stress inside cells. So you can think of all the internal scaffolds of cells as being internal pre-stressed. It has like this internal forces being generated by myosin motors, and it gets transmitted up to cellular scales to adhesions. Okay. And through the wonderful work of molecular cell biology, right, all the hundreds of components in each of these cytoskeletal machines have really been well characterized in terms of their structure and function at the molecular scale. And I guess so for the purposes of this talk, even though I introduced this by thinking about multicellular properties, I'm going to take us back to single cells because as the title of my talk implied, we've been trying to think about how to use artificial intelligence, machine learning techniques to really rethink the way we tackle mechanobiology questions in adherent cells. And my strategy so far for approaching machine learning is I better be working on a project that I really know really well and I have a good gut feeling about, so I know when it's telling me bullshit. Okay, so that's been my approach. Is I need to work with something I know, so I know the mechanics of single cells. So that's what we've been working with. And here, this is a lot going on, but so in single cells, if a single cell is spread out on a glass cover slip, right, you have this beautiful actin-myosin cytoskeleton. That's where all the contractile forces are being generated. The protrusive forces are at the cell periphery. That's how the cell generates new extensions to the front. And the adhesions are these sort of elongated structures that you see around the cell periphery. Okay, so these are these complex physicochemical machines that are controlling the morphology and dynamics at the whole cell scale. And we want to understand how these are integrated from the protein all the way up to the cell scale. Now, we have for 20 years, 25 years, been able to take advances in light microscopy and biophysical measurements to take images. So this is a movie that we took when I first started my group of fluorescently tagged focal adhesion proteins in a migrating, this is an osteosarcoma cell line, a U2S cell, which is a favorite of people who don't like to care too much about their cells, but they're pretty to image. They're really easy to transfect and everything. So what you're looking at here is one focal adhesion protein with a fluorescent protein through spinning disc confocal, and you're watching the cell move, and you're seeing new adhesions appear at the edge of the cell. They change their shape, they disassemble at the cell rear. So these are dynamics that if you're used to looking at doing live cell imaging are just natural to you when you take movies, right? You see all these dynamics. At the same time, this movie was taken by culturing the cell on a very compliant hydrogel. So we could do measurements of the forces that the cell was impinging on the underlying substrate. So this is a technique called traction force microscopy. You basically put fiduciary markers into this soft hydrogel. You measure the displacements, the elastic properties of that underlying gel. So this is a heat map. It doesn't show directionality of those traction stress vectors. All the vectors are balanced, right? The cell is in mechanical force balance, and they're pointing towards the center of the cell. So it's this big contractile stress being applied to the underlying substrate. Okay, so we've been able to take these movies for 2 decades. And the question has always been, what is it about these protein distributions? Can I make a physical model that is built in from our understanding of the protein dynamics? Okay. Oh, this is just a movie about traction forces that I don't think I need to show, but this is the actual schematics that go, like the data that goes into it. We take a whole field of beads, we do image processing to calculate the displacement field, and then back calculate the traction forces. Okay, so one thing that I just want to emphasize, again, I know most of you are not in the field of adhesion or the actin cytoskeleton, is that all these, to be able to enable the dynamics of these adhesive and organelles, is that they're force sensitive. So these assemblies, these cytoskeletal assemblies, change in response to mechanical cues. Okay, so one example is when you take this image of this focal adhesion protein paxillin, and then you use an inhibitor of myosin 2 contractility. you change the morphology of these adhesions, and you also change their biochemical composition, right? At the same time, you can see that the traction stresses go down. So you can do this dynamically back and forth all you want. There's a time scale of rearrangements on the order of 20 minutes, but this is completely reversible. So these cytoskeletal assemblies can assemble and disassemble in response to forces. Now, the same thing happens if you look at the active myosin cytoskeleton. So this is an image of the myosin filaments in a cell like this. It's a different shape. So, but you can see all these nice bundles that we call stress fibers. And so this architecture is what you typically see in some type of highly contractile cells. Okay, it turns out those bundles are not essential for generating contractile force, but it's a common structure you see. Now, if you do the same inhibition, all those bundles disappear. And again, this is reversible back and forth by just on the time scale of 10 minutes. And just to emphasize that, as I can play this, this is actually a movie. Oops. Oh, sorry. Of washing out that inhibitor where you can just watch the self-assembly in minutes and seconds of those bundles and the time scale of 10 minutes, right? So the question is that, like, when I think about all these organelles, not only have they have some complex physicochemical state, but that state changes in response to mechanical force, right? This is quickly becoming a very challenging problem to study, right? And this is just a summary and sort of schematics of the kinds of questions and the challenges that are thinking about when we try to understand dynamic subcellular processes. But you can think of scale this up to dynamic process at tissue scales too, is you have underlying all these structures is a dynamic steady state that is either has a mechanochemical system that's enabling it to maintain homeostasis, right? To keep it in that steady state, or a process, a feedback that's allowing it to drive transitions. So it's this like entire dynamical landscape where all these feedbacks are either allowing you to maintain your current structure or drive a transition. How the heck are we going to figure this out, right? This is a really hard problem. And so, like all of these employ force sensitivities with unknown exact inputs. Like we don't know exactly what the mechanical input is, right? There's all these changes in morphology, mechanics, and biochemical composition of these structures at any given time. Okay. And so, so how we typically approach this. And so this is this example I introduced before of how you go from a biochemical field, that's what you're getting when you're capturing an image, to a physical field, like that's what you're doing when you're measuring a traction force map. How do you do this? Okay, so that's where modeling comes in. And these are the sort of the state-of-the-art approaches one could do, right? And we've tried all of these for this simple, very simple model, right? Let's see, I'll start with the top one, which is sort of like the continuum physics-based approach where you make a very simplistic assumption of the mechanics of the structure. You have 2 or 3 favorite order parameters that you intuit based on your intuition from non-living matter. And then you say, ooh, let's hope a cell behaves like that. And then you test that model and you see if it works. And usually if you contort your cell into different geometries, you try to make your measurements in a certain way, it sometimes does work, but who knows if those parameters are accurate? We have no way of testing it. There's not interpretability. On the other hand, like you might go at the other extreme, right? And say like, oh, if I could only measure every single molecule composition and biochemical rate, then I would know everything, right? And so that's another approach, right? If you, then I could just build everything from the bottom up, okay? And then, if you don't know one rate or you don't measure one rate of something, you have way too many parameters and your system becomes like not constrained at all, right? And then of course there's other intermediates that, pre-modern machine learning, you could say, oh, I want to take some of the information from my images and take some of the information from this microscopic scale and some of the information at the continuum scale, and I'm going to try to find some intermediate. But it was always hard to figure out what microscopic properties to keep in, which ones not to, right? This is a challenge we've probably all been facing if you care about anything above a single molecule. Okay, all right, I see some nodding. Okay, so that's where we were. I think I stopped publishing in this field on this problem about 10 years ago because I was like sort of depressed, right? You can keep doing this and you're like, what am I gonna do? Okay, so not saying we have the answer, but at least solve that midlife crisis. Okay, so this is like a summary of all these issues. It's like, there's so many proteins, there's all these really cool mechanochemical couplings. There's all these different length scales I could choose to focus on. And sometimes, you know, living matter should be different than physical matter. What do I focus on? Okay, so these are all the challenges. And I think what I've come to is I think that there are approaches that we can use modern machine learning to try to advance just the way we approach this problem, right? And I would say that, like, in the examples that I've given, like, oftentimes with those images, because the way we were approaching formulating a model was having the model drive what we as an experimenter were making measurements on. It was this like loop where only a very small amount of the data was being actually used. And so now with machine learning, we can actually take the data, not put our own bias onto what to measure, and let it tell us what we should be focusing on. Okay, so I'm seeing it as like a third partner in a way to build models, and we'll see how far it goes. Okay, so I'm going to try to convince you over the next half hour or so that these are sort of the ways I'm thinking about problems. Some of them are more mature, like we've had one publication in this field earlier this year with our colleagues Vincenzo Vitelli and Patrick Opes and Matthew Schmidt, where we basically did two of these, which was use machine learning to learn what biochemical fields are the best predictor of a state, a physical state that we want to measure. And then use the data and machine learning to then, as a tool to improve the physical model that we build. And these other two are really in more development of how do we build from all the imaging data that we can acquire better ways to characterize all these complex states and state changes that we observe to build statistical descriptions of cellular states and different scales. Okay, and I'm happy, please ask questions during the talk if you have questions. Okay, so during the pandemic, Vincenzo, my colleague, approached me and said, do you have any data? We're interested in trying to see how to use this data to develop neural network approaches for biology. And I said, and we couldn't take any data at that point. So I said, I basically gave him the problem that I posed to you, is like, we have all these images, and there's actually Patrick from Patrick Oakes' lab, all these images of time-lapse movies of protein fields, of cell adhesion molecules, and traction force measurements. Okay, so how do we go from a field of a protein, Zixin in this case, and then to measure a force field? Okay, so Matthew found a neural network architecture where he could train a neural net, this UNet, on images of Zixin, and then minimize the loss on the data, the experimental data of the traction force map, which is the experimental data is down here. And then through iteration and training, then feed the trained neural network. This happened over about 1,000 image pairs or several thousand image pairs. Then give it data and cells that it had never seen. So what I'm actually showing down here is an example that was not in the training set, where you now give a new cell of a brand new geometry, you feed it a Zixin image, you get out a prediction of the machine learning, and then you're like, oh, wow, this is really close to the experimental measurement, okay?
So that's your loss function, is that comparison at the end?
For the training set it is, it's a
Right.
loss function in the
And then
training
you
set,
go through
that's
a
correct.
Series of nonlinear transformations of adding more layers to your network.
Matthew did check all of that, yes, yeah,
Okay.
yeah. And so like the architecture that was pretty important was including the skip connections between before the downsampling and the UNet, because that was a way to allow local information to propagate across the UNet. It wasn't sufficient to have the sort of more traditional UNet. Okay, so I just want to show you how this works. Or how well this worked, which was already surprising to me. So this is an example of a cell that was not in the training set. So this is an example where we have the input, what we're putting into a neural network, the prediction from on a trained neural network, and the experimental data. I also want to emphasize that a portion through this movie, what we're going to do is add in one of those drugs that minimizes the myosin contractile stress. So the traction forces are going to go way down because there's less stress being generated by the cells. And I want to emphasize that the training set only saw cells that were in the wild type condition. So the cells never saw any cells that any data that was really that was at this low stress. Okay. So so what you can see is that, you know, over this whole time course, right, even as the cell changes shape, as the myosin stresses go way down, the neural net is doing a very good job of making accurately reflecting the experimental measurements. Okay, and this is where we washed it out again. Okay, and this really surprised me for a while. I'm just going to tell you why, because it's like, you know, it's way outside the training regime, right? It seems very noisy. You know, for many years, the heterogeneity that's in these images, the fact that every single cell we image is a different shape, there's different curvatures, there's some small adhesions, some big adhesions, was always a pain in the butt when we were doing modeling, right? You always wanted to synchronize your system to have the similar set of conditions. But for neural networks, I think that variability is actually giving you more robustness. So all that heterogeneity that is seen as like the death knell of making any models in biological data, it's actually when you do this on these animal cells, you're starting to get all that variability, and it's using it to train a more robust network. That's the sense I'm getting. Okay, so so then what Matthew was able to do was then we could feed it different types of input data. So here are a series of different images of two of the actin-myosin cytoskeleton, of the actin filaments and myosin, two examples of different focal adhesion proteins, mitochondria, which we didn't think was going to have a good correlation, and then even just the cell perimeter, right? Just the cell mask. Okay. So then we can start to ask how well each of these do. Now, I first want to just start with the easiest one, which is just the cell mask, right? It does a pretty good job of making a prediction of the force field, right? Which gives you sense that the geometry of the cell and the overall shape and its area is already a very a strong predictor of the traction forces, which is, I'll tell you in a few slides, something we had actually known from a continuum model that we had developed about a decade ago. All right. And then the other, so once you think that the geometry is fine, it sort of explains why you might expect all of these to work, is because all of them are giving you some sense of the cell shape. The mitochondria really does the worst of any of them because it has fluorescence signal in places that is not really correlated to the force at all, right? These are these little objects distributed across the cell. And then the actin-myosin do fine, but really the ones that are the best here. So here is really the paxillin and the zixin. So these two focal adhesion markers, because those are actually telling you where the stresses are transmitted to the underlying substrate, right? Which is sort of obvious. Again, I'm trying to start with an obvious example here. And what then we did was go through and look and see whether, because we found that paxillin and zixin were doing the best, we asked, you know, we can take pairs of images. We can do 2-color imaging to ask, does feeding in the neural network images of paxillin and zixin do better than just zixin alone? Or and the answer is no. That really just that zixin as a field by itself is actually doing as good of a job as adding any other additional information, which means that there's something special about that particular protein field that we need to know when making a calculation of the overall force. All right. So at this level, this is a tool that we can use without any interpretation of the neural network of if we're imaging something and we want to make a prediction about something else. And in this case, it was about a force field, but it could be about maybe some dynamics, something in the future, right? You can ask which field of thing that of all the components I could be imaging, which is the most predictive of the thing I care about, right? And so that's a very useful tool as an experimentalist to know what I should start to focus on, okay? So another thing we can do is then look at sort of how much of the size of the cell do we need to look at, all right? So Matthew could do this by looking at the entire cell or just portions of the cell. And what you start to see is that for the smallest box sizes, you know, like these really little areas, you have quite a bit of error. But as soon as you get to this middle intermediate box size, you start to do as well as when you get to the whole much larger size. So it's giving you this idea is giving you a length scale over which you need to train a neural network to be able to get information about the force field again. So you need to have, in this case, I would say, you know, approximately 1/3 of the cell or something like that, right? You need to have enough sense of the geometry. to make predictions of the forces. Okay. This part is actually was motivation to me. It wasn't John, Matthew and Vincenzo are both theoretical physicists, so they're not as much interested in mechanobiology. But this started to make me think about how we could use this trained neural network to really ask about the mechanobiology of adhesions. Okay, so once you have this trained neural network, you can then use it to feed in artificial data sets and ask if I feed it a series of features that I think are reflective of, say, adhesions. Which of those features and which geometries or orientations or densities are most sensitive, are going to be, and intensities are going to give you the affect the force output at those adhesions. So what we could do is screen it, put in all these sort of simulated data sets of sort of artificial cells or artificial focal adhesions, and ask how does that affect the total force at that adhesion plaque site. Right, and what you start to see is that the highest forces, right, that if you screen through different aspect ratios and areas, you start to see this optimal forces happening in this regions, this certain aspect ratios and sizes, which from years of looking at adhesions, we knew that the adhesion size, that sort of basically as adhesions grow, they start out small and round. They grow in area to a certain aspect ratio, but if they grow any further, they start to decrease in force, right? So this type of feature is telling you what morphological information is interesting, again, to think about the mechanics of the force output of that adhesion plaque. And we haven't really thought about how to use this further, right? So like, as another example, once you identify that aspect ratio and area, then you can ask how does the intensity of the protein, which is our simulated cell, affect the overall force output? So now, instead of changing the morphology, we just change the intensity of the white spots there. And you can see that now for different areas, for the smallest adhesions, the force at that adhesion is pretty insensitive to the Zixin intensity, this protein intensity. But as the length gets bigger and bigger, right, now it becomes more sensitive. So for the elongated, larger adhesions, a higher intensity is highly correlated to the force output. Okay, so this kind of exploration of the sort of the link between the local protein density and local force output is something that, again, as an experimentalist, what we often did was just take line scans across our images and try to look for correlations. Now with this trained neural network, we can actually do this in a much bigger sort of statistical set to try to understand these sort of complex connections between biochemical fields and forces. Okay. Yeah. Please.
Yeah. So these little geometries that you're checking here.
Yeah.
The previous slide you showed you get more error as you get smaller and smaller. How big are these features that you're checking relative to the error plot that you were showing on the previous slide? Are these in the?
Oh, okay, sorry. So this error plot is okay. So the plaques that I'm showing right now are individual adhesion plaques.
Okay.
So this area here is a region of a cell.
Yeah, it's a region.
Yeah. So and again, I'm sorry if this is confusing. So the neural network is learning very localized information about that has to do with Zixin intensity, and it's also picking up information about the overall geometry, right? And so right now I'm focusing on what we can learn from the local information.
Yeah. So these the little ellipsoid or ellipses would be in like the smallest box.
The smallest. Yeah, that's right. But what I meant to indicate here is that we cropped our images to be the different sizes and asked how well did was the neural network able to be trained at all?
Oh, okay.
That's what we were asking. Like, do we need to feed it the entire image, the entire cell, or can we feed it different portions of the cell?
Okay.
Okay.
Thank you.
Yeah, of course. Thanks for asking the question. Okay. And as I mentioned, then once you have these, sizes and intensities, then you can ask, how does it depend on density? And so again, just by changing the density of these local plaques, you start to affect the overall intensity, right? And I put these up there just to sort of think about the ways you could use a well-trained neural network to learn about the local predictive power of any field that you're measuring to start out with, right? And I would say even for this, we don't really have we haven't finished our next project on this, right? This was a proof of principle to show that we could use this. Okay, I don't need to show this. Okay, so the next thing I want to show is how we can then use this to improve our ability to build biophysical models. Okay, so 10 years ago, Patrick Oakes, when he was a postdoc in my group, along with Sheila Banerjee, how I get rid of that? Well, hopefully it'll just
That's somebody here. How did they do that?
in the room, you mean?
No,
No.
she's a technical.
Oh, okay. Anyways, I like the thumbs up. Okay. Sheila Banerjee and Christina Marchetti, we built a continuum model of how adherent cells regulate forces. Okay, so again, the observation that we had is that an adherent cell has these sort of localized hotspots at the cell periphery. And in this model, which I'm not going to go through how we tested it, basically all you have to worry about are a few coarse grained parameters are a bulk contractile stress, right? How much contractile, you imagine the whole cell is acting as a contractile sheet, a continuum elastic sheet, right? And that sheet is actually uniformly coupled underneath the cell, right? That's the simplest model, right? There's no localized adhesion. And then we also had to add this term, this line tension term, so we wouldn't get stresses along these elongated regions. At the time, we always felt that this line tension term was a little bit awkward, but we added it because we didn't know what else to do. And I'm just going to tell you right now, we got rid of it in this case, because what we're going to do is put this physics into and constrain our neural net to this physics. Okay, that this simple physics of a contractile uniform sheet. The other thing I'm going to do, so this is this uniform elasticity, this Young's modulus. We're going to make this adhesion dependent on the Zixin intensity. So that's going to be, that eta is going to be like a dependent on the Zixin intensity. Okay. So, sorry, that should have been, okay. So this is what it looks like. So we take this continuum model. This is what for this arbitrary shape cell, this continuum model look like. You see uniform stresses all along the periphery. If you integrated those stresses, you get something that's approximately correct, but it doesn't look right to the data. And now we're going to ask how we could then take information from our data or from the neural network to get something that looks more like our experimental data set. Okay, so as I sort of described before, is that we take this spatially pinning Y that is then dependent on this Zixin intensity. And so when you put this physical bottleneck, is that you have the neural network, and when you you calculate the parameters, and then using this physical model, then you calculate your forces from that, and then your loss function forces you to update the rules of your physics that you're imposing, right? So this is a way of training your neural network that is constrained by the physical laws that you're inputting it. And again, here we picked a simplest physical model that we trusted pretty well, because we had tested it under certain conditions, and we can make the most minor modifications. Okay. And so when you do this, and there's all sorts of problems with doing this, I'll just warn people, is that what you can start to get is an estimation. If you trust this model, you can start to get an estimation of the parameters that you've put into your model. In this case, the most interesting parameter that we could learn is this spatially varying pinning field, or the adhesion field that we get, right? And so we can start to look at how the adhesion active stresses depends on both the Zixin field and then learn the spatially dependent adhesion field. So if you have a physical continuum model with a few parameters, you can constrain your neural network by those physical laws, and then use that to learn the parameters of your system. Okay. We've done this one other time, not in a biological system, but an active fluid. And so we know the active fluid is driven by a hydrodynamic model that's been really well calibrated. What's super cool about that is, again, experimentally, it's often hard to learn your physical parameters and constrain your experimental measurements with some continuum model. And the neural networks allow you to sort of read out these physical parameters from the data itself, right? So I think this is a really great tool to use if you have a continuum model you really like, okay? Now, if you don't have a continuum model you like, there's also ways of just putting in a basis set and learning what Green's functions, what length scales are important in your system, right? So in this sort of Green's function approach, you can have your trained neural network, you can learn your sources, but then you have this layer of Green's functions that then helps you predict your fields, your force fields, and learn what mathematical expressions relate those to a force field. So here it's equivalent to like saying like you have a series of point charges, right, in space, and you want to learn what electromagnetism is, right? That's the exact same type of approach we're doing, is that we have a series of variables and we want to learn what those fields are that are controlling the variables that we're measuring. All right, so this again is all math. There's no physics in it, right? And the Green's function is very confusing. But the thing what's really interesting about this approach is if you look at the Green's functions, what it does is it learns two different fields. One field which is very localized, right? This pink field, this pink Green's function, and it can be explained by these two functions, right? The pink one falls off very sharply. That is at a length scale equivalent to that of a single adhesion site. That's that very local parameter. And the yellow curve is a more long length scale decay, right? And that is, we think, something more analogous to that elasticity parameter that we think. It's sort of like a long, I mean, you can even think of hydrodynamics or elasticity, but that's that long field parameter, right? So what it says is that it is reasonable to build a model, a physics model, with two different length scales in it, one that would be very local and one that would be long length scale. Again, for full disclosure, right, I said I didn't trust machine learning, right? So I'm doing this with problems that I sort of knew the intuition about beforehand, and it makes sense that this was what comes out of it, right? Okay. And what's sort of impressive is even with this very simplest description of the Green's functions, right, you have this this prediction from just this simple Green's functions neural network that does a reasonable job of predicting the force field, right? Even with all the constraints and the mathematical constraints and only having two Green's functions there, it still does a pretty good job of predicting the force field, right? So I think this is really exciting to think about how you could start to bridge different scales from your imaging data of how you can make larger length scale predictions from these localized measurements. So this sort of pipeline, and this should be updated, this is what was published in Cell earlier this year, is like we sort of identified these three different approaches to thinking about how to build data-driven pipelines for biophysical modeling, right? This first it's just this question of if you have two fields or you have a dynamic field that's changing in time, like, using uninterpretable neural network just to see which fields are important for making future predictions, I think is already a very powerful tool for experimentalists. And it helps you sort of identify which fields are important, and then also start to learn what rules, if you put the physics that you think is right into that system, or discover new sort of new mathematical relationships between the fields that you might not have thought about given the physics. Clearly, we would like to get to a point where we trust this approach enough that you could say learn the mathematical expressions and then learn new physical rules that allow you to understand how these systems are adapting and evolving. But I don't feel comfortable saying we're there yet. Okay. Okay. And another way of just like sort of visualizing this sort of different model is that, like, there's all these different, if you think about experimental data being on this, like, it's an actual measurement, but you don't know how to interpret it, to a continuum model where you know exactly how to interpret it, but you don't know if it's connected to real life, right? There's like this nice axis of interpretability between this, where you can use, let's say, deep neural networks, which is hard to interpret, but at least it does a good job of prediction. And maybe for some of our experiments, that's totally fine. Like, I don't actually care about understanding. I just want to make a prediction for something that I care about. But if you want to go between that and some interpretability, you can start to have machine learning as a tool to aid your sort of physics constrained interpretability of those neural networks. Okay, so this is something we're very excited about extending upon to other types of problems. All right. Okay, so that's this first section of sort of just thinking about how do you actually use was our first foray into like thinking of what information is in the biological imaging data sets that we haven't used previously, right? There's tons of data that's there, and we don't. How do we take advantage of that? The last couple minutes, I'm just going to spend. It's even more preliminary. It's stuff that we've really started since that other paper stopped, but or like since that was published. And it's really thinking about now that we have this view of statistical learning as a tool, what else could we do with it, right? And so this is me brainstorming up here, and I'd love to have feedback from you. So one thing is that we're trying right now is sort of tackling this question of all the information content that's in the images that we don't take advantage of. I heard earlier say that segmentation is a challenge. Yeah, we hate segmenting too. But even with the segmentation is like when you have all these segmented features and they all have different shapes and they have different maybe mechanics and different biochemical compositions, what do you do with it? All right, so here we're doing this in the context of focal adhesions where we can do immunostaining on all these different biochemical markers of different focal adhesion states, do image segmentation of individual focal adhesions, and then do an unsupervised cluster analysis of all the different adhesions and see if we can learn what different kinds of adhesions exist in the cell, right? So again, us as experimenters have sort of characterized like nascent, new adhesions, mature adhesions, fibrillar adhesions, and we've sort of assumed a function for them, but can we actually do this in an unsupervised way so we can actually learn what those representations are? And so we've just started doing that with a computer scientist, computer vision expert in the center. And she's been able to see different clusters of adhesions come out. And then once we have that unsupervised clustering, those could be then inputs into like a larger neural network of the adherent cells, right? Okay. The other thing that we're really thinking about is transitions, right? And so if you can characterize a single cell state, and it doesn't matter what scale, it could be a state of a single focal adhesion, could be state of an entire cell at some stage of the cell cycle. How do you learn about those transitions in the cell state? Okay, how do you know when a cell is going to enter a next stage of a cell cycle, or a focal adhesion is going to change things, right? So, so this, we want to think about these state transitions using these statistical approaches that I described before, but then start to think about how they're constrained by physical rules, right? So, I like to say that like when we think about these sort of abstract notions of a biochemical and physical state of the cell, when they make transitions, the living matter is actually physical stuff, right? It has to be obeying laws of physics at all these different scales. So this has to then, we need to put this into our understanding of how these transitions are occurring, right? And so here, this is something I've, put some, if we understood how to characterize these states and then make these transitions, this would allow us better tools to be able to engineer living systems and make these state transitions. Okay, so I'll just tell you the best example that is now embarrassingly old and then got scooped by another group, which I'm sort of annoyed about, but I'll just tell you about it anyways because I think it's cool. So, we asked again about 4 years ago this question of like, if you look at a collection of epithelial cells, this is time-lapse movie of, cell membrane staining. And so you can see cells going through the cell cycle, the cells are changing shape. And we can measure the cell cycle by using a reporter of the cell cycle state, a Fuji reporter. So up here is experimental measurements, cataloged whether the cell's in G1S or G2. Okay, so at that time, 4 or 5 years ago, we wanted to ask the question is if we just take a movie of the cell shape, could I make a prediction of the cell cycle that it was in? And the answer, and does it care what cells it's next to? Like, is there any information on that's in there? And the answer was shockingly, well, to us at that time, you could. All you need to know was actually the size and the shape of the cell, and you can make a using sort of a neural network approach, like your neural network would make a very good, accurate prediction of what stage of the cell cycle you're in, okay? We now know that because it turns out that in these epithelial cells that the shape of the cell is and the size of it is actually a good predictor of whether the cells are resting in G1. But there was actually now 2 papers out sort of showing the same thing, both in epithelia with cell shape. But then there's another paper that just came out that if you only do like DAPI staining on a cell, like just look at nuclear staining, you could actually train a neural network to make predictions of the cell cycle. All right, so this idea that how much information you actually need to be capturing and how much is captured in morphology, I think is like, there's just going to be a lot of progress made on this, and neural networks are a very useful tool to know sort of like what's the minimal information that you need to make predictions about something that's way more, what you think is way more complicated, right? So if you want to know your cell cycle, maybe all you need to do is do a DAPI state, right? Like that's sort of amazing, right? Okay. I have one more example that, what happened to the image? Oh, here we go. I just want to show one more example. And this is something that's actually funded by the BioHub up in Chicago, is another kind of state transition we've been thinking about is in endothelium, this transition between a sort of a anti-inflammatory and pro-inflammatory state that can occur from different kinds of like flow cues. So if you have different kinds of flow profiles in your endothelium, or if you have different agonists of inflammation, right, you can drive changes in cell morphology, but also barrier, so cell-cell barrier that really make the system pro-inflammatory, okay? So now this process of going between something we consider anti-inflammatory and pro-inflammatory, if you ask any given person, there is lots of different ways you could characterize these different states, right? So you could look at changes in gene expression, you could look at the secreted factors, you could look at the cell surface receptors, you could also look at the morphology, right? Each of these different types of hallmarks of inflammation is something that you might choose to characterize changes to pro-inflammation. Now, our question is pretty simple, is like if you just add an agonist to it, do all these markers change at the same time? Now, I think we'd all agree that they're probably not going to change at the same time, but if you had an unbiased way to characterize what the state transition looked like, you could start to see the pathways in which this inflammatory state transitions, whether all the inflammatory states are similar to each other, and or what is most predictive of whether, like say in this case of inflammation, if you're going to be able to recover from an inflammatory agonist, or you're going to just stay in this hyper inflamed state, okay? So this is sort of a quantitative way of how we're going to start to address these different questions of cell state transitions. Okay, so I did listen to your guidance a little bit. So, all right, that's what my science, that's sort of what we're doing. And I did want to give you a little bit, especially on one touch point of what our NSF Center is doing up at Chicago. This is a physics frontier center, so it's much smaller scale than what you guys are launching here. And so, you know, our center here involves 18 different faculty members at UChicago, and so it's, you know, founded on research. And I mean, roughly speaking, the 3 focus areas of the whole focus of the center is understanding how living systems adapt and learn and retain information across scales, right? So this, the first research area is really about molecular evolution, like how do you build molecules that are set up to adapt? So Arvind Murugan and Michael Rust are leading that effort on molecular evolution. The second MA, which is really about morphogenesis, is really thinking about how cells and tissues change their shape in response to stimuli and control their shape. And the third area is sort of taking ideas from neuroscience, like that's sort of classic of how biological systems learn and process information, and applying these ideas from neuroscience to other areas of learning and information processing at the scale of ecological scales and also inflammation. So we sort of are organized in this way, but might see there's lots of synergies. I thought the synergy with this center that one thing that we're trying to do is take some of these ideas of how to incorporate machine learning into building better biophysical models, and specifically in getting computer vision tools that are necessary to do this, and also high throughput sort of synthesis approaches. Okay, so these are two approaches that were sort of developed. I talked a lot about the use of machine learning and AI and imaging data. Rama Raghunathan and others have really talked about when you look at create large libraries, right, and you see variation, can you use this as a way to guide new design, so evolutionary inspired design? So getting the protocol set up to be able to make these types of assays more accessible to others. And so in the area for the imaging thrust, this is the one that is definitely the most developed here. We've hired a staff scientist, Leah Ding, who started about 10 months ago. She came from the Allen Institute for Cell Science and then worked at a neuroscience institute. So she has been working to help us with computer vision techniques, right? And this death valley between, people who like to take images and people who like to make models. This is where my lab always lives, is like no one ever wants to build the image analysis tools. I mean, we do, but it's definitely hard. I mean, it's getting harder with all the 3D imaging modalities, right? So that's something that I think could be a good point of synergy with here. Like if there's, we're really and also interpreting those images. And we're mostly focused on getting that started right now. But we're generally very interested in helping to build AI into better models for quantitative biology and biological physics. Okay, so thank you for your attention. The people who did the most of the work on the project I discussed was Patrick Oakes, who's now at Loyola University, Matthew Schmidt, who's a senior graduate student with Vincenzo Vitelli, my colleague in physics, and then Shailaja has done a lot of the work with endothelial cells. And the last thing I just want to say is that if you aren't aware of it already, there is a National Institute for Theory, Mathematics, and Biology up in Chicago. It's a joint Chicago Northwestern endeavor. They host workshops often, and it's really encouraged for participants there. You can submit a proposal for workshops, right? So it's a great resource in the Chicago area that we should be taking advantage of. Okay.
Is that connected at all to the SACOM node that's in Chicago?
No. No, it's an NSF Simons Center.
Oh, okay.
Yeah, it's actually run by Rich Carthew at Northwest. I mean, it's run, Northwestern was the primary.
Simons, okay.
That is a Simons Center, yeah.
Right, well, thank you very much.
Yeah.
Several questions.
Hi, you go.
Hi, so, you said perhaps probably tongue in cheek that, whatever the cell cost or HGK, you don't care about the cell, but presumably you look here, so how do you validate or how do you infer that that those very artifactual cells, their biology do reflect the way these things come together in real life, in organism.
Yeah. I mean, I would say that that's 25 years of focal adhesion mechanobiology. You know what I'm saying? Like, I mean, those particular results, I mean, it's just the whole field works on different cells and you see which things are common. I mean, I think that's how that, for focal adhesion mechanobiology, that's how that was done.
Let's say,
Yeah.
as an example, how high or what's the most realistic? So does one see that in, let's say, developing mouse embryo, fly embryo? What's the realist biology that one observes?
Focal adhesions or
Same characteristic from the bigger characteristic as one characteristic of
I mean,
not just the observation, you say, but sort of support that everything we infer about the mechanism is relevant.
Yeah.
It's relevant.
Do you believe structural biology? No, I mean, I guess my feeling has been like, when you think about cytoskeletal, yeah. That's right, yeah. Yeah. No, I mean, with cytoskeletal assemblies, right, these are subcellular organelles, right? So I think when you think about macromolecular assemblies at that scale, right, those forces and those properties that are gonna drive, macromolecular assemblies are gonna exist across scales, right? Now, you can see with these biophysical measurements, you can't do many of these measurements in developing animals, right? But you can take some of the ideas, and there's been drugs developed to affect integrin activity, at the molecular scale. Now, I would say that like part of the issue with say trying to tackle metastasis, right? Like something that involves migration and the complex morphological changes, like being able to tackle morphological dynamics that aren't tackled by a single molecule perturbation is still a challenge, right? That's a challenge, like those diseases are very hard to tackle, right? Like if you can't tackle it with a single molecular target. I mean, I'm not answering your question. Well, I think that in terms of testing, like, the field tests it by looking at in lots of different contexts, right? And then seeing certain genes and certain structures in animals, but you can't do the same kinds of measurements there,
Just as an example, the two-dimensional kind of almost nature of many of those.
Yeah,
It's cultured cells.
yeah,
Don't think that speaking from the physics point,
Yeah.
you don't think that is, let's say,
artifact? No, I mean,
Right, but something that have a fundamentally different behavior in the three-dimensional.
Yeah, not at the scale of adhesion assembly. I mean, cell morphology is gonna be affected. I mean, and also you do see changes in focal adhesion size. That was like an argument for like 10 years in the field. But like, yeah, no, I don't think the mechanobiology at that scale of the organelle is gonna be altered. I mean, the whole cell does affect the mechanobiology, right? I'm not gonna argue that. So like, if you have a cell that's epithelial, the epithelial cells that are in tissue are gonna be different than a mesenchymal cell that's spread out on glass. Yeah, and a mesenchymal cell on a soft collagen gel is gonna be very different than that, right? But you can take the ideas that are learned from mechanobiology in these different contexts and they link up to it, like it's not, yeah.
Yeah,
Yeah, so really cool stuff. So you showed us these pictures where you used the network to predict the mechanical
Yeah.
behavior of a cell. I was curious if you take now surface with patterned elastic moduli, you can actually potentially offer surfaces that look like that.
Yeah.
And then see if the real cells, when they adhere to such a surface, actually also respond in this way.
I see, yeah, so just enforce the adhesion geometry and see that,
Exactly.
yeah, exactly.
But
So
that's
that,
something that's
honestly,
present.
that was sort of what we built all our models on
Yeah.
the first place, but we haven't gone back and carefully checked it with the neural networks. Yeah,
But
that's
I've never
yeah,
seen anything, I mean, like these
that's right.
highly
Yeah,
symmetric
yeah,
surfaces, I've
exactly.
never seen anything like
Yeah,
that.
yeah, and there's all sorts of things that we should be doing to break that neural network. I mean, we could imagine doing it, yeah.
Yeah, I was sort of shocked at how few parameters you really needed to do that. Because I remember when the Paul Allen Institute published that image on their stem cell, they had labeled several proteins and all the different organelles.
Yeah,
And then to watch the stem cell grow and divide.
yeah,
And, they were just putting like, I think they had to label 20 things.
yeah,
And then put the time trace for each one of those
Yeah.
together.
so I mean,
that was
we're,
an inspiration already back in
yeah.
2018, I think.
Yeah, we're doing okay, so we're like, in terms of the focal adhesions, we're like doing the stupidest type of prediction. We're trying to ask, not in time, just like at an instance in time, can this protein field predict a mechanical field, right? And so that, in that case, we have lots of sort of mechanobiology insights as to why Zixin is, which I haven't had time to get into, but we know it's a force-sensitive protein that we know why, we have a good reason to think that why Zixin is very good. Okay, so that's just, yeah,
Label.
just with GFP tagging. I mean, this was, yeah,
Because it's, something's nice, at the end, they were hoping to do it label free, so
Yeah.
first you get the information from the label. But
yeah,
I think as all of us have slowly learned, you label anything in the cell, like we know from the minimal cell, it
Yeah.
changes its doubling time slightly
yeah,
to very large.
yeah,
So, but I think it's a brilliant idea, and when you look at these, particularly these time evolving images, they're so complicated to try and correlate so much information.
that's right.
That's
Yeah.
amazing. think that would be a really good use to have that at the back end of a microscope.
I think so too. Yeah, I think, or even in as a tool that experimentalists use. I mean, yeah, I do think it's important because I think going forward, I think a way I could do it, like we did spend years in the lab taking images of, say, the actin cytoskeleton, right? And trying to look for correlations between actin and forces, because I thought that it was a mechanical structure. It should be important, but its architecture is actually not very important. And it took us, 7 years to understand that. With this, we could have understood it a lot faster, right? To know which fields are predictive and which ones aren't. I do think that's a useful, yeah.
Any other, oh, Martin?
How much redundancy is there? Since by just a little else, so we can say.
Yeah.
Just randomly.
Wait, sorry, what did you say?
So how much redundancy is there? Since if I take these ellipsoids.
Yeah. Yeah,
What parts of the force?
that's a great question.
How many?
Yeah, it's a fantastic question. Yeah, that's a, I don't know the answer to that, but it
would be like an ideal system to actually.
yeah, no, it would be.
Just create experimental.
Yeah, I mean, our neural network, if we believe that, I mean, says that there isn't much redundancy because the density is actually pretty sensitive. And so there is something about density. I still don't quite understand what that is, because I mean, I have some more hints because of ongoing work in the lab, because we think all these, I mean, this is probably going too much, but I was actually talking, I think you're a student, about this, like all these Zixin molecules are basically little force sensors on the actin cytoskeleton. Like there's a whole class of proteins that are acting like force sensors. And we think they're feeding back into mechanochemical circuits to alter both Rho activity, so the contractility, and also the repairing the actin cytoskeleton. So the intensity is actually sort of a readout of the mechanical state of the actin cytoskeleton there. That's sort of an input to these mechanochemical circuits. Does that make sense?
Yeah.
So I have a model of this, but has not been done precisely. Yeah.
Maybe then actually.
It would be so cool. Yeah, I would love to do that. Yeah.
So if there are no further questions, let's thank Margaret one more time.